Okay, welcome back, Wayne County Community College students. We're now going to start the final video for digestion. We're in the physiology now of the digestive tract or the digestive system. We've gone through the anatomy. You know everything that's involved in the anatomy, from the mouth to the esophagus, to the stomach, the small intestines, large intestines, and out the anus. So make sure you know all those areas, and the flexures for the hepatic flexure, the splenic flexure, know which side of the body those are on. And remember the cecum and what hangs off the cecum, the appendix, and then go over all of your abdominal organs that we discussed. Let's get to the physiology now. I have some words on this board to kind of familiarize you with. I'm sure you know them by now because you've been studying, and the fact that you already did your lab assignment, so I know you know these words already. But I'm going to go over them anyway. Ingestion. This is the intake of food or materials. That's what ingestion is, simple. Digestion. Digestion is the breakdown of complex nutrients into simpler nutrients. Digestion actually begins immediately after food has been ingested. The process of chemically and mechanically breaking complex foods into simpler nutrients that can be absorbed, more easily absorbed, would be a good definition for digestion. Motility. Motility is the muscular movement along the GI tract. That would be an example of peristalsis. You're going to learn about peristalsis and segmentation, but remember, segmentation just breaks things up into smaller components as a to and fro, back and forth action. Actually, we're going to introduce that in this section, so I want you to pay attention to the differences between peristalsis and segmentation. Yes, food does get moved along in that way, a little segmentation, a little bit of peristalsis, a little segmentation, a little peristalsis, but peristalsis is the major way that we have motility in the GI tract. Secretion. Secretion is the release of digestive juices. Digestive juices are just a one big category to have enzymes, acids, bases, mucus, bile, etc. So we just call that whole concoction digestive juices. And this will release these juices into the lumen of the GI tract. So when we talk about secretion and absorption, you have to know which direction things are going. Not only for the GI tract, but you're going to see that too when we get to the kidney. Which way are things coming back to the body will be absorption, and which way things are going to be secreted. That will be going away from the body or becoming waste that we eliminate. In this case, this would end up going into the toilet. So absorption is the movement through the GI mucosa into the internal environment. This is when you want to absorb your vitamins, you want to absorb your minerals. The main reason we eat is to fuel our cells, cells, to fuel our cells, right? To make sure we have enough nutrients to keep ourselves going, to make sure we stay healthy. This is why you have to opt to have more fruits and more vegetables in your diet. For sure, it keeps things moving along your GI tract, but it's also the way that we get nutrients into our cells, right? And for sure, drink enough water. Elimination. Elimination is excreted digestive residuals. You could call it defecation, you could call it stool, you could call it poop, feces. People have a few other choice words for it. I'm going to leave those off because you get the picture of what elimination is. And then we have chyme. Chyme is a mixture of partly or partially digested food and digestive secretions that are found in the stomach and the small intestines during digestion of a meal. So a mixture of this partly digested food and secretions, enzymes, and stuff that are in the stomach and the small intestines during the digestion of a meal. It will consist of a thick liquid. And of course, the color will vary because it's going to depend on what you've eaten. Say, for example, you ate a lot of kale or collard greens or a lot of spinach, then that kind will be indicative of that by being a kind of greenish color, right? So let me go over these two, secretion and absorption, so we'll have a good idea about what that is, and then we'll continue on with the digestion. Let's just go over this real briefly so you can understand it. Here's just say this is the tube of the GI tract, right? When things come into it, let's see. Let's say when things come in this way, that is secretion. You go inside the lumen, release of digestive juices, enzymes, acid, bases, mucus into the lumen of the GI tract. So things are coming into it. Remember, if you secrete, you excrete. This is the stuff that's going to stay in the tract and end up going into the toilet. That's the way I always remember it. On the other hand, for absorption is the stuff that when you have eaten a good meal, or even if you eat a bad meal, this is what you're going to absorb. That's why people say you are what you eat, because if you eat a healthy meal, you're going to absorb healthy things, good vitamins, nutrients, etc. You eat terribly, always going to McDonald's, then that's called junk food because you don't really get the nutrients that you need. And that will be something that comes out of the lumen and into our bodies, into the other cells. So this is absorption. So absorption is going to come out of the lumen and go into our bodies to feed our other cells. Secretion is going to go into the lumen and eventually into the toilet. So absorption, movement through the GI mucosa into the internal environment, which would be us, our cells. So I want you to kind of rhyme secretion with excretion, and you'll remember that whatever being secreted is going to stay within this lumen and eventually go into the toilet as excrement. And whatever you absorb, absorption is coming out of that lumen and coming back into the body. And that will be a way for us to get the vitamins and minerals that we need to stay healthy. All right, so digestion begins immediately after food has been eaten or ingested. The process of chemically and mechanically breaking complex foods into simpler nutrients that can be easily absorbed would be a great definition for digestion. Let's look at a couple of ways we digest food. One is mechanical. We're going to look at mechanical and chemical digestion, but let's first talk about mechanical digestion. Mechanical digestion breaks large chunks into small bites, or small bits. It consists of all movable or all movement, motility of the digestive tract, and it brings about the following: the change in the physical state of ingested food, churning of the stomach contents, the stomach churns, propelling food forward along the digestive tract would be the three things that occur during mechanical digestion, right? When you think about mechanical digestion, think of like a machine, something mechanical. That will be chewing, that will be the peristalsis of getting the chyme and the foodstuffs through a system, the GI system. And then what about all those muscle layers, the longitudinal, circular, and oblique muscle layers in the stomach that could cause food to churn sideways, diagonally, and up and down. So that's a nice churning, a rotating of the food stuff. So that's all mechanical. Anything that deals with seems like machinery or movement will have to do with mechanical digestion. Let's talk about mastication. Mastication, I've used this word before, and I mentioned to you before that we were going to come back to it. And here it is now. Mastication means chewing. Chewing. Mechanical digestion begins in the mouth when the particle size of ingested food material is reduced by chewing movements or mastication. Also, mastication serves to mix food with saliva in preparation for swallowing. So that leads us to another word, swallowing. Deglutition. Right? Swallowing is the movement, the moving of a food bolus. A bolus is like when you are chewing or eating your food, you typically turn it into a ball, a little ball, and then we swallow it. Okay? And this is moving a food bolus from the mouth to the stomach. You have an oral stage from the mouth to the oral pharynx. Remember, oral pharynx means mouth, throat. That's under voluntary control. You can control what you chew and whether you move that food source towards the back of the throat, known as the oral pharynx. The pharyngeal stage is from the oral pharynx to the esophagus. At this stage, it's pretty much involuntary. We don't have much control over that. As we begin to swallow, the food starts to go down the esophagus, and we consider that involuntary control. Although you do know there are portions in the esophagus that are voluntary, but that's very much up in the upper 1/3 position of the esophagus where the striated muscle is. Then the final stage of deglutition or of swallowing would be the esophageal stage. This is from the esophagus to the stomach. And this is definitely involuntary because now the food is leaving the esophagus, going through what sphincter? Right, the lower esophageal sphincter opens up, the LSE, correct? Lower esophageal sphincter, and then it goes on, the food stuff goes on to the stomach. Let's look at peristalsis and segmentation. Okay, peristalsis and segmentation. Peristalsis and segmentation. After food enters the lower 1/3 of the esophagus, this is where smooth muscle is. This is usual using either peristalsis or segmentation. These are the things that are responsible for the motility or the movement of the foods along the esophagus. Peristalsis, though, this is a wave-like ripple of a muscle layer, of the muscle layer of a hollow organ, initiated by a bolus stretch. So the food comes in and reflexes are there. The hollow organ knows to kind of open up and start to move things along. This is peristalsis. initiated by a bolus stretch, causing a contraction of circular muscle. So when I think of peristalsis, I like to think of movement. When I think of peristalsis, I think of movement, right? Segmentation, on the other hand, it's movement, but just kind of mixing. Well, we could say it's mixing movement, but not really movement along the track. But it's a mixing movement. It's a back and forth, a to and fro type of thing. And this back and forth action breaks up chunks of food and mixes the digestive juices, right? So when I think of segmentation, I'm going to think more so of mixing. And I'm going to probably ask a question on the exam about these two. So when you think about peristalsis, think of movement. You think of segmentation, think of mixing. You can always go in the text and read more. Of course, I encourage you to do that. Always supplement your notes, because my notes are mainly just so you could get the big picture. But little details are within the text and are within the readings that I've assigned. So when you have peristalsis and segmentation, when peristalsis and segmentation occur in an alternating sequence, food is churned and mixed as it slowly progresses along the GI tract. So here you have movement with peristalsis, it's moving along, but here the segmentation is breaking everything down to and from. If you think about peristalsis, maybe you could think about it as when you have a toothpaste tube, and at the very bottom, you're trying to squeeze some more toothpaste out, or even when you are trying to squeeze toothpaste out, even if you squeeze it in the middle, it doesn't have to be the very bottom. But that kind of action, that squeezing there, propels the toothpaste out of the tube, right? Then you squeeze it, toothpaste jerks out, and you can squeeze it, toothpaste comes out. So you can squeeze it in the middle, you can squeeze it in the bottom, wherever, and the toothpaste will come out. That's like peristalsis. A little bit of squeeze in the, say, the esophagus, and then the food moves along. A little bit of squeeze, food moves along. So they have a great picture in your textbook about that. The regulation of motility. Let's see. So please note that it takes about 2 to 6 hours after a meal to empty the stomach. So it takes a minute. It takes 2 to 6 hours to empty the stomach after a meal. Of course, this is going to depend on the amount of food that was eaten and definitely the contents of that meal. Some people say like certain ethnic foods just go straight through them, so they don't feel that fullness for very long. So it's going to depend on how much you've eaten and the contents of a meal in order for the food to go from that 2 to 6 hours. Food is churned with gastric juices, and they form a thick, milky material known as chyme. I think we introduced chyme before. Chyme. Right? This is really kind of some mushed up food. Kind of thick, and it's going to depend on the color of the foodstuffs you've eaten. This chyme is then injected into the stomach, from the stomach, excuse me. This chyme is then ejected from the stomach into the duodenum or duodenum, and it goes in there approximately every 20 seconds. Since the stomach is considerably larger than the duodenum, this pyloric sphincter must open and allow some foodstuffs to come in and then close. So it's opening and closing. It lets just a bit of chyme come in to the duodenum, because the duodenum is very small. It's about 10 inches, if you recall. C-shaped, 10-inch little first part of the small intestine. So to regulate how much content, food content comes, or chyme comes in from the stomach into the duodenum, this pyloric sphincter is there to prevent overburdening the duodenum. We have some regulatory hormones. Gastric emptying, to empty the stomach, is regulated by hormonal and nervous systems. The GIP, gastric inhibitory peptide, this is a hormone that's released in the presence of fats and carbohydrates into the duodenum, or I should say the presence of fats and carbohydrates that are in the duodenum, so they're in the duodenum. It decreases peristalsis in the stomach and slows down the passage of food from the stomach into the duodenum. So gastric emptying, gastric inhibitory peptide, gastric meaning stomach, inhibitory means it's going to stop. This peptide, which is a protein, this is going to really slow down gastric emptying. It's going to slow down peristalsis, right? Another one, nervous control. It uses receptors in the duodenum that are sensitive in the presence of acid and distension. This is called the enterogastric reflex. The enterogastric reflex causes a reflex inhibition of gastric peristalsis in the vagus nerve. Now remember, the vagus nerve, you're able to digest food and really, it's when things are slowed down. And digestion can occur when your nervous system is kind of slowed down. The vagus nerve helps put a little brake on things, right? You know, like it's that whole thing with the parasympathetic nervous system. So the vagus nerve will slow things down with this hormonal control, or with this nervous control of the enterogastric reflex, which will also is a reflex that inhibits gastric peristalsis, inhibits and slows down the emptying of the stomach as well. So far we got hormonal control of emptying the stomach, and we have nervous control. Intestinal motility includes both peristalsis and segmentation. Peristalsis is regulated in part by the intrinsic stretch reflexes caused by this bolus of food, this ball of food that's in the mouth, going down the esophagus, etc. It will stimulate the peristalsis, this movement, right? Also stimulated by the hormone is cholecystokinin pancreazymin. We call it CCK, cholecystokinin, when you see that. But some authors do put on this pancreazymin, which I hadn't seen before. But please note that we just really call it CCK. This is secreted by endocrine cells of the intestinal mucosa whenever chyme is present. When chyme is present, CCK is released. Only during a digestive period is CCK released. This is a digestive hormone released during the time of eating. Only during a digestive period. You do not see CCK when there's a fast. Okay, just keep that in mind. CCK release is evoked by the presence of chyme. So you got to have some foodstuffs around. So of course you've eaten. In the duodenum. So you got chyme in this little C-shaped duodenum, and it induces the contraction of the gallbladder. And I think I mentioned that when we were looking at the liver and the gallbladder. The gallbladder is stimulated by CCK. So that's definitely in a fed state. And this is cholecystokinin. You can see it in your text and look it up. And even though they put the word pancreazymin in it or on it, that doesn't mean anything. Just look at the CCK and you know that is a hormone that stimulates the gallbladder to contract. only seen during the time of a fed state when you have eaten. Now let's look. So let's now turn our attention to the chemical digestion. We talked about mechanical digestion. Talked about the way food and chyme moves through our system. Now let's look at chemical digestion. Chemical digestion consists of all the changes in chemical composition that food undergoes in the digestive tract. And this is a result from food hydrolysis. Hydrolysis is a chemical process in which a compound unites with water and then splits into simpler compounds. This is seen in the action of the enzymes in the gastric juices. So we got chemical digestion. Now before, mechanical digestion had to do with like machinery, the chewing, the churning in the stomach, the peristalsis in the esophagus, and then the rest of the GI tract. Now this is chemical. Anytime we talk about chemical, we're going to talk about, put some enzymes in there, some pH, some bile, some acids, some things like that, some bases. So these are all things that are chemically induced. And we have hydrolysis with this chemical digestion. So that would be, let's look at the properties of digestive enzymes. These are part of the chemical digestion. So the properties of digestive enzymes. I think we've talked about enzymes before, but if we haven't, I think we have. But let's just go over that, review it. All enzymes are specific. They're specific in their action. That is, they act only on a specific substrate. They're very intentional, enzymes are. Digestive enzymes function optimally at a specific pH and become inactive if this deviates beyond its narrow limits. So digestive enzymes are very specific, and if anything comes and interferes with their activity, mess up their pH, then they won't function anymore because they're very pH specific. Changing the pH changes the shape of an enzyme molecule and may make it inactive. So that's how important it is. So here you have, so I need you to remember that enzymes are specific. Any change in their pH can make that enzyme inactive. So enzymes act like a specific key to a lock or a hand to a glove. Very specific. So let's just draw maybe a substrate, and I'll just make the substrate like this. So that's the substrate. And the substrate is usually the thing that the enzyme is going to attach to, so to speak. And here's the enzyme. It has to come in and fit it specifically and perfectly. And we don't care what this part looks like, but this part where it's coming in to fit, that must be on point. So here's the substrate, here's the enzyme. They must fit at this point. This is the way a lot of receptors are in our bodies, and that's why we can have so much going on in our bodies at one time. Like certain chemicals will bind to certain receptors, even now, certain viruses will bind to certain receptors in our body, depending on what it looks like on the phospholipid bilayer, right, on the outside of our cells. So I want you to remember about substrates and enzymes, very specific, and particularly digestive enzymes. Changing the pH of them can cause a great effect. So let's look at, now we're going to go into all the different types of digestive enzymes. Let's take a look first at protein digestion. Protein digestion. You have very large molecules made up of hundreds of folded or twisted chains of amino acids. Proteases, you see the word protein, and aces, that's another thing I wanted to mention about enzymes, is that usually, not always, because we're going to meet two of them that don't, but usually enzymes have the suffix or end in ace. Usually, but we are going to learn about two, I think they're protein enzymes that do not. But let's just say for in general, the enzymes do end in ace, have a suffix of ace. Let's go on. So these proteases, even though I want you to use context clues, you're on the exam and you see this word and it asks you, does this enzyme, and you know it's an enzyme because it ends in ace, right? In this particular case. Oh, do these break up fats, carbohydrates, or what? Proteins. You may not know the word, but you have to always look for context clues. And if you see a word that's got most of the word, like protein in it, please mark that it breaks up proteins, even though you may not have ever seen that word. Always look for context clues. You don't have to always know everything verbatim. You could just figure it out. You can use deductive reasoning or inductive reasoning. So here we have proteases that catalyze the hydrolysis of proteins and into peptides. Ultimately, the protein digestion is going to break up these proteins into amino acids. And the peptidases in the intestinal brush border. So these proteins are in the intestinal brush border. They're going to break up the proteins until they get to amino acids. Please remember that I need you to know all of the end products of proteins, of fats, or of lipids, of carbohydrates. Look those up and know the end products of those. So here we have two different enzymes. Still talking about protein digestion here. And we'll see how this pans out. So we have, still in protein digestion, we have pepsin. Pepsin is a protein digesting enzyme. Yes, I know it doesn't end in ace. We have two protein digesting enzymes, but neither one of them end in ace, but most enzymes do. But anyway, here's pepsin. It is a protein digesting enzyme in the gastric juice. It functions optimally in a low pH range. Now, when we have a low pH range, would you say that is acidic or basic? A low pH range is acidic. Good. So you have a low pH, and this pepsin works optimally in a low pH, so that means an acidic environment. Environment. Let's see. So if you had an acidic environment, what area would you say is the most acidic environment in our bodies? Good. So this would be found, this enzyme would be found in gastric juice, in the stomach, of course. That's gastric, that means stomach, juice. It will be inactive. And we said if you change the pH, that makes for digestion, it makes the enzyme inactive. It will be inactive without, remember that S with the line over it means without hydrochloric acid. Hydrochloric, or we'll just say, or acid. But in our stomachs, it's hydrochloric acid. So that's the one we mentioned, HCl, hydrochloric. So pepsin is an enzyme that loves a low pH. It functions optimally in a low pH environment. A low pH environment means it's acidic. Where's the most acidic or acidic environment? In our digestive tract, because that's what we're talking about, it will be the stomach. If you don't have an acidic environment, maybe you took something to neutralize all the acid, like an antacid in your stomach, then pepsin isn't going to work very well to digest proteins. Let's look at another protein digesting enzyme. Also, it does not end in ACE, but these are exceptions. Trypsin. Trypsin is another protein digesting enzyme, and it functions optimally in a higher pH range. It likes a high pH range. So if your pH is higher, would you say that's an acidic environment or an alkaline basic environment? You got a high pH. Good. High pH means you're in a basic environment. And you know you can use basic or alkaline, means the same thing, environment. Now, what in the GI tract, this is what we're talking about, where is alkalinity? Where is basicity? I mentioned this to you before. Where does a base come in to neutralize all that acid that's coming from the stomach as everything enters into the duodenum? Because everything has to go into the duodenum. Yes, the pancreas. Good. So this functions optimally in a high pH range. And so we have this word, chymotrypsin. We're not going to trip over this just because they added the word chymo to it. We can see a word we know and understand as trypsin. We just said it's an enzyme, so don't trip just because they add chymo to it. Chymotrypsin is in pancreatic juice. The pancreas is alkaline, it is basic, and it will help neutralize the acidic environment coming from the stomach. Both of these are protein enzymes. Protein enzymes. So, if I had, let's see here. Suppose I had a little chart, and this is the protein, and this is the pH here, and we say that's 0, 1, 2. And I have protein from here to a protein to here. So just say we have your proteins on here, right? And the pH. So this hump here is obviously more in the acidic range, because here's the pH 0 to about 6 here for this graph here. This is protein versus pH. Always look at your graph and look at the X and Y to see what you're reading in the graph. Which enzyme would this be? It functions optimally in an acidic, low pH environment. Correct. That would be pepsin. So this would indicate pepsin. On the other hand, here's a protein. Here's an enzyme that loves to be in the more higher regions of pH. like about 5, 6, 7, 8, 9, 10, right? It's a little overlapping here, but we won't worry about that. But overall, it likes to be down on the graph in the higher regions where the pH is higher, it's more basic, more alkaline. And that would correspond to the enzyme secreted from the pancreas. That would be trypsin, would be that enzyme. Both protein digesting enzymes, but they act different. The pH is very important to these enzymes in the gastric juice, in the digestive system. pH is very important. Okay, so let's look at carbohydrates next. Because remember, we can only digest proteins, carbohydrates, and fats. So we're going through, and let's see how we digest carbohydrates now. Well, carbohydrates are interesting in that they start off really kind of big, we could say, with polysaccharides. But let's just write the word carbohydrate. So carbohydrate digestion, they call it CHO for short. Okay, so carbohydrates are saccharide compounds. Make sure I got all this paper. They're saccharide compounds. Let me just write that word. So, yeah, polysaccharides. These are sugars, by the way. Then you have disaccharides. And then we have monosaccharides. Okay? Now, when we have polysaccharides, really, that's like starches, glycogen. They contain many in this group. So polysaccharides, these are many sugars, like several sugars, right? Poly meaning many, contain many of the saccharide groups. Disaccharides are two sugars, two saccharides, right? Okay. And then mono will be one saccharide. This is the one we're really concerned with because this is the one we can digest, monosaccharides. But let's go through them. So a polysaccharide would be starch or glycogen, right? Okay, so that's gonna be too big for us to really utilize. So right away, we need an enzyme here to break this up into disaccharide. And the one that we have directly in our mouths, in our saliva, is amylase. Amylase. Now I'm going to need you to look that enzyme up and learn a little bit about it because I have 2 questions on the exam about amylase. Amylase is in our saliva naturally, and amylase is also found in the pancreatic juice. I need you to understand where the pH of this ranges and how it affects breaking up polysaccharides like a potato, for example. That's a nice starch into a disaccharide. So amylase will break up a polysaccharide into a disaccharide, and it will hydrolyze the polysaccharides to disaccharides. It's found in the saliva and in the pancreatic juice. Now, we get to the disaccharides. They're going to need some enzymes as well to get it to be a monosaccharide. These are some enzymes, 3 enzymes we could use. You could use sucrase, You can use maltase, and you can use lactase. These are the enzymes we're going to use, right? Okay. So these enzymes break up sucrose, maltose, and the sugar lactose, right? So you look at sucrose, you know that's table sugar, and you know that lactose is dairy or milk, sugar found in milk and ice cream, etc. Cheese, milk, and we'll just leave it at milk. This is an ice cream too, but so if anybody is lacking this enzyme, I'll talk about this one specifically. If anybody is lacking this enzyme, lactase, we call them lactose intolerant, meaning they don't have the enzyme to properly break up this disaccharide into monosaccharides. And usually that leads to being kind of gassy and bloating and an uncomfortable feeling. Well, of course now milk has, you have lactose intolerant milk or lactose-free milk, lactose-free ice cream, etc. So they just take it out of the, they just take it right out of the beverage now, or whatever it is you want to eat. So these sugars actually contain two, let me just see if I can get that. So sucrase are two sugars, these are disaccharides. So this is really not working, that's what it is. Okay. This is really glucose. So sucrose is made up of glucose and fructose. Maltose is made up of glucose and glucose, just the way they're bound together. And then lactose is made up of glucose and galactose. Okay? You can see there are two sugars. These are disaccharides because that means two. Right? And these are the enzymes that are needed to break up these two sugars. Why? Because the goal is to get to a monosaccharide. That's really the goal. We really, really want to get to use this glucose. So now we're down at monosaccharide, glucose. Although any of these are monosaccharides, the brain and the cells really, really crave glucose more than the other monosaccharides. All of these are fructose, glucose, and galactose are monosaccharides, but glucose is the main one. And that is the one that is our end product of carbohydrate digestion. Very easily absorbed. So this is easily absorbed. And this is the one our brain craves, right? That's why you don't want to go on a no carb diet, because then you're depriving your brain of the very energy that it needs and desires. Plus the fact you can tell when you're around somebody who's not eating carbs because they're evil as hell. All right, so let us now, I want you to remember that, what are this? Enzymes, sucrose, no, they correspond to that segment. And these bind at the surface of the brush border. Please know that is contact digestion. Binding at the surface of the brush border. Binding at the surface of the brush border. This is contact digestion. I think I have a question on that. So don't ignore that when you see it. Contact digestion. And we saw what a brush border looks like because I drew it with the microvilli, and the villi looks like a little brush. Okay, so that is carbohydrate digestion. A couple of things I want to mention about that before we move on is the amylase, which we mentioned here. Amylase is an enzyme that hydrolyzes polysaccharides into disaccharides. It's found in the saliva and pancreatic juice. It's the main enzyme in saliva. It functions best in the neutral to slightly acid pH range. And that's characteristic of saliva, to be neutral to slightly acidic. I think I'm going to ask a question about that in the exam. The enzymes sucrase, lactase, and maltase catalyze the final steps of carbohydrate digestion. So these enzymes catalyze the final steps to carbohydrate digestion. In other words, going from a disaccharide to a monosaccharide. I want you to note that they correspond to the disaccharide substrate and bind at the surface of the brush border, and this is known as contact digestion. So these fit perfectly: sucrose, sucrase, enzyme, substrate. Enzyme maltase fits perfectly into substrate maltose. Enzyme lactase fits perfectly into the substrate of lactose. The end product of digestion for carbohydrate digestion is glucose, which is easily absorbed. Most enzymes catalyze a chemical reaction in both directions, but not digestive enzymes. So you may see digestive enzymes, they only catalyze reaction in one direction and one direction only, not both directions. So it catalyzes in one direction and one direction only. Let's take a look at fat digestion now. That will be interesting to see how fats are digested. Let's take a look at fat digestion. Almost finished. All right. Now this is fat digestion. All right. Fats, as you know, are insoluble in water. In other words, they don't dissolve in water, so they are insoluble in water, right? Fats have to be emulsified. In other words, they have to be broken up into very small fat droplets before they can start to become digested. So lecithin and bile salts, both found in bile, emulsifies fat in the small intestines by breaking these large fat drops into small drops. So you got a big globule like this. Well, then bile can come along to this fat globule or glob. Bile can come along, act on it, and then you can get these smaller fat droplets known as micelles. Okay? Now we're a lot more prone to being able to absorb something like this. This would be pretty difficult. Bile acts like a soap to fat. Have you ever seen like those Dawn commercials where they'll have this greasy pan in the sink? Usually it's an empty pan of lasagna. Then they'll have water in it and they'll squirt the little Dawn in it and you see all the fat kind of dissipate. Well, that's what bile does. It breaks up fat so that we can actually start to digest it better. Makes it easier for the pancreatic lipases, that's fat lipids, lipases. So if you saw And this word, you'll know that it breaks up fat. I want you to know though, because I'm going to ask this bile is not an enzyme. It is not an enzyme. Okay? This is an enzyme. The only other two that we looked at that didn't end in ACE, there were enzymes, was the ones were the ones with the protein, pepsin and trypsin. This bile is not an enzyme, although it does help break up fats from big old fat droplets, globules, to little fat droplets. It breaks up fat globules to smaller fat droplets. It is not an enzyme. Lipase, and I'm going to ask about some other enzymes, but look for the ACE on those, and you'll know that they are enzymes. Please note that this lipase, and this makes it easier it comes from the pancreas. Pancreas does indeed help break up fats, and you saw the pancreas also helps break up digest protein, right? Okay, so the pancreas is involved for sure as a digestive enzyme. This is the main fat digestive enzyme that works. It comes from the pancreas. Bile is produced by the liver, but is stored in the gallbladder. Bile is produced by the liver, is stored and concentrated in the gallbladder, meaning when it's concentrated, water is taken out of it. It's released into the lumen, so it's going inside the GI tract through the common bile duct. So make sure you go over those ducts that I drew when we learned about the gallbladder, and you'll remember. There's some residuals to digestion. Cellulose, for one. Cellulose is a dietary fiber. It's a carbohydrate, actually. Collagen is another residual of digestion. This is like undigested connective tissue, right? Cellulose and collagen remain undigested in humans because there's no enzyme to digest them. Some fats can be bound to calcium and magnesium to make the fats undigestible. I don't know if you remember this one a while ago called Olestra. Olestra, they would make this, this was a fat that was bound to either calcium or magnesium or something, and you can't digest it, so it's like you can eat all these chips and stuff and things with fat, not snacks, and not have the weight gain because you're not digesting the fat. People end up so bloated and gassy they were miserable, so they had to take that off the market. I want you to know that these undigested wastes, bacteria, pigments, water, and mucus are what make up feces. So you got undigested wastes, bacteria, pigments, water, and mucus is what makes up feces. Cellulose is like, grass. So if we were at WC3, you know how the lawn looks. It's a beautiful lawn there. You could put cows and horses out there. They could eat the grass and get really huge because this is a carbohydrate. But we could go out there and eat the grass and nothing will happen because we don't digest cellulose. Now, I said that for years, and then I read in some journal, scientific journal, that people that are indigenous to Japan, they actually have developed an enzyme in their gut to digest cellulose because they eat so much seaweed. So, but when they come over here and say one generation, they will lose that ability to digest cellulose. So they are able, if they stay on that island and that's what they eat, a lot of seaweed and stuff, they have developed, I guess it would be cellulase. I guess that would be the enzyme that would help them digest that, the seaweed and the, I think it's stuff they put in like sushi or something. So that may be something. So I used to say humans don't digest that, but I did read something about that. And then collagen is just, have you ever had like the bone part, the little gristle part of the, like a chicken bone? We could chew on that and eat that, but we won't digest that. so that collagen will not be digested. So that's undigested connective tissue, and this is a dietary fiber. Cellulose is carbohydrate. One last thing, I want you to know about the control of pancreatic secretion. Secretin, I want you to know about secretin and CCK. All right? We talked about it, but if you could just go into a little more depth in your text, that would be great. So look at the control of pancreatic secretion. Right? So you got this word, it looks like secretion, but it's not, it's secretin. Secretin is not secretion, it's secretin. And CCK, which is cystokinin. I think your text may have put pancreozymin in here. We don't really call it that, but help you remember that it is an enzyme. So secretin here, this evokes the production of pancreatic fluid, low in enzyme content, but high in bicarb. So it's low enzyme, but high bicarb. I'll just put bicarbonate. So bicarbonate, if you see bicarbonate, which we will learn about when we get to the kidney, that is going to neutralize acids. So it's coming from the pancreas, so you know it's going to be an alkaline substance, so it will neutralize acids. Here, CCK, cholecystokinin pancreozymin, was just the opposite. It causes the increase of exocrine secretions high in enzymes. All right, so it's high in enzymes. It causes the pancreas to increase exocrine secretions, high enzyme content. It opposes the influence of gastrin. So gastrin, when gastrin is released, if you recall, it releases a lot of hydrogen, HCl, right? And therefore no HCl stimulates the contraction of the gallbladder so the bile can pass into the duodenum. So when we talk about how do we get a real good bile going into the duodenum, please remember it's CCK. Although both of these can do it, CCK is the main one that will cause ejection of bile from the gallbladder. So secretin and CCK can do that, but like I said, put your money on CCK because that's the one everybody always asks about. I think for this lecture, I'm just going to flip through here, make sure I talked about everything, and I think that's pretty much it. If you have any questions, you can feel free to email me, you know that, and then, I'll try to answer it the best I can. But this gives you a pretty much overall view of the lecture. This is the end of the lecture on digestion. We did the anatomy portion. This was more like the physiology portion. Feel free to go through the text, especially look at the pictures and read the captions under the pictures. Another thing I want to mention to you is that glucose is passively exported out of the lumen of the GI tract by co-transport. So glucose has to be hooked up with sodium to be transported out of the GI lumen. And you will remember what a lumen is. That's the inner part of the walls of a vessel or an organ, right? That's the lumen. So remember to look up co-transport And really, if there's anything else, I think overall you got a pretty good picture of what GI is about. And I will talk to you later.